Hey, how's it going, guys? My name is Samuel Myung, and today I will go over the Nirenberg and Matthaei experiment along with the Nirenberg and Leder experiment. Let's go. So Marshall Nirenberg and J. Heinrich Matthaei started off with a poly-U, which is a homopolymer and essentially a long string of uracil nucleotides. They added the poly-U to a test tube that had a cell-free translation system, 19 unlabeled amino acids, and 1 radioactively labeled amino acid. This test tube was then incubated for translation to occur. Once this had all been done, they filtered out the protein and the filter was checked for radioactivity. They knew if the protein was radioactive, then the labeled radioactive amino acid must be in the protein and the codon codes for that specific amino acid. They then ran this parallel procedure in 20 tubes, with each tube containing a different labeled amino acid. They saw from their results that the tube with the radioactively labeled protein also contained freshly synthesized protein, with the amino acid being specified by the homopolymer used. In this first trial, they could only make a protein with phenylalanine. Therefore, they found that UUU specified the amino acid phenylalanine. They were able to also identify that CCC encodes proline and AAA encodes lysine. They unfortunately could never get GGG to work, no matter what methods they used. Eventually, Nirenberg partnered up with Philip Leder to determine other matches that existed between codons and amino acids. They went with a triplet binding assay where they synthesized short mRNAs with already known codons and then added it to a mixture of ribosomes and tRNAs with amino acids. This allowed binding to occur where the ribosome in the mixture was then able to bind the mRNA and the tRNA specified by the mRNA. They next passed the mixture through a nitrocellulose filter where the tRNAs coupled with ribosome-bound mRNA got caught on the filter versus the unbound tRNAs that were able to pass right through the filter. They then checked the filter to determine which amino acid was bound. For example, when an mRNA with GUU was used, Nirenberg and Leder found that the tRNAs on the filter were bound to valine. Therefore, they could conclude that the codon GUU encodes valine. Using this method, they were able to identify many other codons and ended up identifying 61 codons. This led to the cracking of the genetic code and the creation of the coding dictionary.